All right, guys, so this is the video to wrap up our notes since we didn't get through everything. We got a little sidetracked talking about David Goodsell and how awesome he is. We also, I took forever to pass out those pencils and things. Just can't quite remember to get that planned in advance. So we started here just to recap. This pinky red area is the nucleus, which is where our DNA is, and our DNA is in the form of chromatin. Chromatin is the DNA wrapped around the histone. So I outlined that a little bit for you. You've got intermediate filaments here helping to support the nucleus. You've got the nuclear envelope with a nuclear pore that RNA would come out of. Over here in our light blue, we have our microfilaments, and we have 3 microfilaments on this top panel, and we've got many, a lot of stuff on my desk, sorry. We've got many down here on the bottom. Continuing back to the top, these orange blobs, these were our ribosomes. We've got one that's free floating here, and then we have lots that are attached to our endoplasmic reticulum, okay? And then here is more endoplasmic reticulum. I did forget to color him in class, so you might want to add him in. He is part of, see the ribosome attached, so he is a endoplasmic reticulum. And then here we have, this is an end of an endoplasmic reticulum budding off. So he is forming a vesicle. So I'm going to go ahead and start coloring my vesicles. I'm going to color them dark green. So this is regular green. So this is my vesicle here. And then he continues, oh, I don't know if that picture of me will be in the video. So here he is right here, and then he continues on the other side down here. Remember, we're going to cut these out and tape them together. So I'm going to label this guy here a vesicle. We'll write about vesicles in a minute. Oh, I can't spell that. That's a C, and then this one is an E. Vesicle. And then we have, this is a vesicle in here. I'm going to skip him. He is got these weird shaped protein structures around him. This is called a Clathrin ring. That's really high level biology. We're not going to go into that. But if you were studying if you continue to go into biology and study a higher level, you will see those, learn about those. So in here is another vesicle. I feel like I need to do my speed coloring, but I'm going to color the inside a little bit lighter. And then here is actually, this looks like an entire piece of the edge of the cell, but this guy here, this is a vesicle, okay? So this is the beginning of exocytosis. So we want to make sure we understand that this is a vesicle. And all of those pieces are about to go into the cell, and out of the cell, sorry. That is a vesicle. All right, so now we need to go back to our Golgi, and we had started writing about the Golgi in our notebook. So here's your Golgi here, your pancake level. And remember, I told you in class, if you wanted to color the inside, you can. I actually have decided I do want to color the inside. Okay, so there is our Golgi. So our Golgi, what we want to write in our notes for our Golgi is that, first of all, it's a Golgi apparatus or a Golgi body. It does have 2 names. I don't care which one. You do need to be able to recognize both, though. We talked in class about the fact that it packages the proteins that will leave the cell. So it's packaging them in a vesicle, and so that they can survive out of the cell. And then it has two sides, and this information is very brand new to you this year. One side, the side that's closer to the ER, so this is the side that the vesicle will come to from the rough ER, that's called the cis face, C-I-S, cis face. And then the other side of the Golgi, and that's the side of the Golgi where the vesicle will leave from to go to the plasma membrane, that's called the trans face. Those are two words that it kind of spaced mine close together. So the cis face and the trans face. And I do want to get that labeled on our picture. So this is our cis face, and then this is going to be our trans face. Okay, so a cis face and a trans face. All right, so the next thing we want to, we need to write our vesicle information down. Cis and trans. So our vesicles are for storage and transport. They fuse with membranes, and those could be membranes of organelles or membranes of, this is too dark. Yeah, this is going to be too dark to cover, so I'm just going to kind of bracket it, box it. I'm going to box it. That's what I'm going to do. There you go. Those are our vesicles. Oops. There you go. Storage and transport, and they fuse with membranes. All right. And then our next structure is our third cytoskeleton structure, and that's this big fat guy. Okay, so here's our big fat guy. And this guy is called a microtubule. Okay, and you see he's the biggest of all three of the cytoskeleton components. And if you remember from your structures in your textbook, he is made of tubes that also form a tube. So he's like a bunch of little tubes that that are arranged together to form a big tube. So there's our only microtubule in this. I use brown for that. It's not as pretty. And then here we're going to write our microtubule. I'm also going to bracket him because I think the brown will not show up as well. We'll cover up my words. So this is the largest of the cytoskeleton. And it is made of smaller tubes that are called tubulins. Isn't that so cute? So tubulin. And this is a track that is also for movement. Okay, so this is a track for the vesicles to move along. Kinesin is the main protein that moves the vesicles along. I'll add that using, because we like him, right? He was so cute, kinesin. All right, I did go up here and I want to modify. So it is the microfilaments, I misspoke. The cell movement that microfilaments help with is the actual contraction of the cell, and with cytokinesis itself. So that is what helps divide the cells into two when they are dividing in mitosis. All right, so our last, the last color that we need to use, I am going to use purple, and we're going to focus on the cell membrane here at the end. So this area is the cell membrane, and right now we have a vesicle fusing with that cell membrane in order for exocytosis to happen, and the contents of that vesicle can leave the cell. Okay, so this is cytoplasm, this guy here, this is the cell membrane. Okay, and this is the outside of the cell if you wanted to add that into your label. All right, so now let's write what we're going to, our information for the cell membrane. I wrote it up at the top of my page because I'd run out of space at the bottom and I didn't want just one little piece of information to be on the back. You could also, if you have not written anything on your other page, you could write this down there when you adhere your picture. So this is cell membrane. The cell membrane is a phospholipid bilayer, and it has proteins embedded in it. Okay, so that's really all we need to focus on for what the cell membrane is. Remember that cell membranes let things in and out. Some things like CO2 might be small enough to get through the cell membrane on its own. Things that are larger or charged may need a protein channel to get through. And then even larger things or bulk transport, we would need endocytosis to bring it into the cell or exocytosis to bring it out of the cell. And that's, of course, the big focus for lab number 2 and for chapter 5. So let's see how, let's see how we put this guy together. All right, so the first thing I'm going to do is I'm going to cut on the little cut dotted line here. All right, so that's what I did. I just cut that in half. This is your top piece. So I am going to just trim the edge up here. You can't see what I'm doing. I'm just trimming the edge up here, excuse me, trimming that off. Okay, and then there's a lot of extra space up here. I am going to get rid of that. That's up to you whether you want to get rid of it or not. Okay, see, there we go. All right, now I wrote pretty close to the edge there, that dotted line, so there's no way to really cut that any closer. And I am going to leave this because it'll give a little bit of extra support to the paper when I tape the other one on top. All right, so for this one, I am going to need to trim here. And I overcut a little bit, not on purpose. I did much better last year. All right, and then you're going to just line them up, and you see they fit almost perfectly. Mine doesn't fit quite perfectly because I messed up the cuts. I'm just going to take a piece of tape. Oh, I moved them. There we go. Tape there. A little piece of tape at the top. This is where if you wanted to continue writing out reticulum, you could, or you could just leave it. I am also going to trim a little bit here on the bottom, just because I'm kind of a perfectionist. And I know I'm cutting off the face words, so I'm going to have to go back and add those in. Okay, so see, I cut off the cis and the trans, where the word face. So I need to get my light green. Thought I might be doing that. Face and face. All right, so I'm just fixing where I cut those words off. And I think I might try and cut a little bit at the top off as well. There's this little overlap here. I don't know, that might bother me, it might not. I'm also going to tape the back here to make it a little bit more secure. I have a bunch of double-sided tape that I'm trying to get rid of, so I'm actually going to tape this underneath. If I didn't have all this double-sided tape, I would just put a regular piece of tape there. All right, so I'm going to try and trim up a little bit here so that it is a little bit more even, at least for a little bit ways. This is just my weird. Okay, and then I'm going to trim this part off. All right, so now what do we do? All right, let's get under this document camera a little bit better. Okay, so here we have our blank side. Let me zoom out. Focus. Okay, I didn't need to write anything, so I'm going to put it right here in the middle. You could also fold it. Ooh, I've never done it this way. I think I might put it this way. But anyway, whatever you're going to do, you're going to put your glue or your double-sided tape if you're like me. Just going to put it on the back here. Okay. And I usually only start with just a little bit. Okay. Line it up with where you want it to go. I'm going to put it right here at the bottom. All right, so now it's nice and it's on there, it's attached. And then I'm going to fold it to where I want it to be at the top of the page. Okay, nice. I'm going to lift this up. I'm going to put some tape underneath. Anybody else like stick their tongue out when you're doing stuff like this? Like, I'm so weird. All right. All right. So now I have folded that. I'm going to line it up a little bit better with my crease. Okay. And then I'm just going to accordion fold back. There you go. And look at that. My title is almost perfect on the page. Okay. So now I have my two pages right next to each other. I've got my tour of a human cell that I can fold out. And I've got my definitions and my key parts and key right next to it. Okay. All right, guys. Thank you so much. See you in class.